So I guess the most important place to start is for the general public to redefine what role protein plays in the human body, because a lot of people think about protein as, oh, that thing that you get from Steak Shack. No, not Steak Shack. Smoothie King, right? Okay. Something that you take when you want to make muscles, right? And that's the general public kind of just thinks about proteins in those terms. But proteins really are the molecular robots of the body. Right, so they are the tiny nanobots that actually make things happen. Right, so of course, one of the things they make happen are muscles, which a lot of people care about. But proteins are also enzymes. They are transport channel membranes. They form antibodies. They put anything that needs to be done in the body. A protein is usually involved. It's a small little robot. So that's the first sort of maybe paradigm shifting thing that most people who are not familiar with biology or proteins in general need to think about. The second piece of the puzzle here is that proteins have a very complex three-dimensional structure. It's very convoluted and complex, and the shape of that 3D structure is integral to the function of the protein. So if the protein were to be a different shape, we call that a denatured protein, which, as the name suggests, is probably not a protein that does a good job. Right. So when we heat up our eggs, for example, and the egg yolk, the white of the egg becomes white, that is because protein is being denatured. It's kind of being destroyed in a way from its biological function. So that three-dimensional structure is of critical importance. The problem is that it has been exceedingly difficult for us to be able to determine what that 3D structure is. We have the 3D structures mapped out for about a few 10,000 proteins. I think the number is 20 or 30,000 proteins. The way that we've done that, though, is through very expensive and complex imaging technologies like 3D electron microscopy or crystallography, which are laborious and time-consuming, and you don't always get it right anyway. Right. So from so that's one building block of understanding this issue, is that the 3D structure is important and that we have not been traditionally very good at determining what that 3D structure is. Now, a slight tangent here is talking about something called the primary structure of a protein. So a protein is basically just a chain of subcomponents. These subcomponents are called amino acids. So if you string a whole bunch of these amino acids together, and there can be in the hundreds and the thousands of amino acids strung together, that string of amino acids basically is called a primary structure. Proteins don't actually ever exist in the primary structure. That's just a nice label that we use to discuss the morphology of proteins. As soon as those amino acids are strung together inside of the cell when the protein is being synthesized, almost immediately, almost instantaneously, that protein turns into what's called the tertiary structure, which is the final three-dimensional structure. So the way this incredibly long string, it could be a long string or a short string, but typically complex proteins are very long strings of amino acids. The way that string, you can literally think of it as a string, turns into that complex three-dimensional structure in this origami-style method, the number of ways that can happen, that can bound together, is literally larger than the number of atoms in the universe. It's almost infinite how many different ways it can fold. And so that's why it is, and that is the proposition was in the 70s that if you knew the primary structure, if you just knew the sequence of amino acids, you could theoretically figure out what the tertiary structure should be. That was the proposition proposed in the 60s or 70s, I believe it was. And so then the race began to solve the so-called protein folding problem, that this is the primary structure. How do we compute or figure out what the tertiary structure is from the primary information? And it was relatively trivial to figure out what the primary structure was. What is the sequence of amino acids? What was not trivial at all, what was a huge problem for 50 years, was to figure out how this primary structure turned into the three-dimensional structure. And this was the so-called protein folding problem. For about 10 or 15 years now, there has been some computational advances. There has been some mathematics developed to figure out that, okay, if we know that these are how certain amino acids bound together, if we know that these are certain motifs and patterns of how proteins fold, then maybe we can figure out computationally, we can compute what it is. So we don't have to do the expensive crystallography, we don't have to do the 3D microscopy stuff. We can just compute it, that it's essentially just a computing problem. So in 1994, a competition was started called the CASP competition, I suppose, and the role of it was to basically. They already knew what a 3D structure of a protein was. They said, okay, we've mapped a 3D structure. We're going to release the primary sequence, the amino acid chain, to whoever wants to participate. And they have 2 or 3 weeks, I think it was, to compute what the 3D structure would be. And this was an open competition. Like, here it is, here's a primary structure. Figure out what the 3D protein structure is going to be. And, whoever gets the closest to the 3D structure wins. And historically, and they have a grading system to figure out how close you got to the 3D structure. And historically, I believe that the median closeness, so quote unquote closeness, that people really got over the last 2 or 3 decades was like 40 to 50%. They were not that, I mean, a lot of the times they pretty much botched, half of, it was 50% accurate at best. And then in 2018, along comes a DeepMind, and they create a AI algorithm they called it Alphafold. And in 2018, they decided, this is a perfect artificial intelligence problem. If this is just a computation problem that requires large amount of data to be ingested and then pops out an answer, well, we may know a thing or two about how to do that. Mm-hmm. So in 2018, they for the first time participated in the competition, and they actually came out on top. They, I think that year they got a score of 60%, which was still substantially higher than any of the other, competitors. And the competitors are all universities, And then this year, they submitted 5. The competition was 5 different primary sequences that you have to compute the tertiary sequences for, tertiary structures for 5 different proteins. And 5 out of 5, Alphafold scored above 90%, which is the highest that anyone has ever scored. And a somewhat arbitrary definition has been created to say that if you get it about 90% right, according to our scoring system, then we will have considered the computational problem of protein folding to have been solved. And so this is, the first time since the proposition was put forward in the 60s that anyone has been able to come up with a successful computational model for protein folding. And that's sort of where we stand right now. So having said that, how significant in your mind is this? Is this just, so should we be excited at all, or?
Yeah, I think it's tremendously significant. Actually, so let me respond to it. I mean, there's a lot of points. I think one important point is why the tertiary structure is important, right? So, you mentioned there's these amino acids. They come together in a string, right? Each there's like 20 different subtypes of amino acids, right? They come together in a string, and then that string folds, right? And making a very rough, not really accurate analogy, if you're 3D printing something, you have a string, but how that string folds determines what you've made, right? The string itself may be the same, but if you fold it into a boat, then it's a boat, and if you fold it into something else, then it's something else. And in particular, because these proteins act as catalysts for reactions, right? The shape of a particular protein may allow, for instance, just simple example, glucose to bind in a particular way, and then that glucose molecule to be broken in a particular way, right? So because of the shape, that's what allows, it's sort of the traditional thing is a lock and key, right? Is the protein forms a lock, and the molecule it's acting on forms a key, and because their shapes match, that is what allows it to do the robotic thing that it does. That you talked about the nanobot concept, but that nanobot is shape based fundamentally, right? Antibodies on everybody's mind as well right now, right? And what an antibody is it is a lock that fits the key of a virus or some part of a virus, right? And so when we respond to the COVID-19 vaccination by creating antibodies against the spike protein, we are creating a protein, the antibody. An antibody is a protein that has a shape that corresponds well enough to connect with that spike protein, right? So that's why the shape in 3D is so tremendously important, is because all of the nanobot activities are shape-based activities, right? And to be even more specific, within that lock, there are little points of attachment that all have to line up with the points of attachment on the so it's the shape, but it's also the shape of those little chemical binding spots, right? And so all of those things have to be brought into a particular 3D orientation in order to effectively bind that and not bind other things. Right. Because otherwise you're doing things you don't want to do, right? So that's it's the shape is so critically important. And then the other just minor point is, and just trying to relate to things that may be already familiar, right? So we're familiar with DNA and the famous double helix, right? So that's also a sequence of base pairs, right? And that helix shape is what they call a secondary structure, right? So if you just talk about the sequence of base pairs, right? But then when it forms that 3D helix, that's a secondary structure. When that helix twists on itself and forms into a chromosome, that's the tertiary structure, right? And the same thing happens with proteins. But the whole point of DNA folding is just to protect it from outside influence. So it just kind of compresses into compact structures that are surrounded and protected. The proteins actually have to do stuff, right? And so the DNA, in our previous episode, DNA makes RNA, RNA makes protein. And the way it does is by creating this string. That string is known to fold in a particular way. And because it folds that way, it does certain actions reliably. And that's the other point is I mentioned secondary structure. Proteins also have secondary structure. So there'll be certain areas in a protein that do form helixes. There will be other structures that naturally form little sheets or what they call beta sheets or whatever. And that was actually critical in Alphafold's solving, because they didn't try to resolve stuff that was known, right? Is that they took existing results that said this sequence of string creates a beta sheet, typically. This sequence creates a helix, right? Alpha helix, right? So I'm not going to try and rederive all that. We're just taking those as known pieces and figuring out how they're put together, right? The other thing I found really fascinating was, and this is something I think that's underappreciated about where AI stands in a lot of ways, is the way that the engineers reduce protein folding to a computational problem is not trivial, right? There's a huge amount of human insight to say, okay, well, what we can do is we can solve for the angles between the amino acids and the probability that a particular amino acid is close to another amino acid somewhere distant on the string, right? And those probabilities combined, the angle plus the distance probability, will give us an idea of the 3D structure. But that's actually quite an amazing insight, that you can reduce the problem down to computing those two factors. And then they use things like their image, the existing Google image recognition technology and all of those things, of those methods, to try to compute these probabilities. But the insight that was the necessary computation to do is actually not trivial, and is actually brilliant, really. I mean, I was pretty impressed with that insight.
I don't know that was AlphaFold's contribution necessarily, because
the distance was.
The distance thing was? I do know that they were recognizing certain patterns already computationally, for a decade or more, so they kind of built on that already.
The phi and psi angles, I think, was known that was going to be part of it. The distance thing had been done by others only as a binary. Are they close or not? Yes or no. One of the primary and what Alphafold seems to, because they chose that computation of distance, not binary, but how close on a continuum, seems to be what has allowed them to get to this level of accuracy, right? So anyway, regardless, I think it's important to realize that the engineering or the computer science insight as to what problem should be fed to the AI in order to generate this result is actually really brilliant. But it's a team. It's the Alphafold team that kind of like reduce the. I mean, you take a complex problem and then you reduce it down into the key elements, and then you put it in. And if it is right, as it turned out to be, then it means you've correctly reduced the problem, right? So anyway, I think that's super exciting in terms of...
and the solution is probably going to be scalable, right? Because, they're going to work backwards on, there's something called a protein database where there's tens of thousands of proteins that have already been mapped out, so to speak, and then they'll run... And, they use that data to create Alphafold anyway. But now they can, sort of run it forward again. They're like, all right, we already know the structure of this. How good is this really, right?
Correct.
And they will continue to hone. And I think now that they've kind of, quote, cracked the code, figured out what algorithm works, I think it will very quickly start to approach greater levels of accuracy very quickly.
I think that's probably true. I think
we're not another 10 years away from almost perfecting it. I think we may be months away. I mean, it sounds crazy to say that. Remember, for AlphaGo, they did the same thing. They created the algorithm. They're like, all right, this is going to beat humans, and then they had AlphaGo play itself, and then it turned into some super godlike Go player. I don't even think they released those games. I don't think they didn't want to freak out humanity with what happens if AlphaGo plays with itself. But there'll be some kind of analogous iteration that they're going to do where the prediction for protein folding just becomes even more granular.
Yeah, and it should be scalable. I mean, the question in my mind, as I was listening to them lecture on kind of their solution, is, so I don't know if you they actually cropped it into segments, the chains, and then basically tried to solve the segments separately to reduce the computational demand. Right. And I think the interesting thing about that is that it worked, because it's not obvious that would work. Right. That may not have led to a solution, but they tried it on a huge database. It worked throughout the database and got better and better. Right. And it was able to solve the problem. And so even when they cut it wrong, I think that's the important part to realize, is that because it's a neural net, it converges on the correct solution, even if you start it in the wrong place. Right. And so even though they often would crop incorrectly or not incorrectly, but in a non-useful way, the program gradually corrected itself and converged on the correct solution. So that's also, I mean, a lot of what they did was not just feeding it to the AI, but tuning the AI so that it was doable. The result was achievable without requiring, 4 months of TensorFlow, this sea of servers, right? You can do it in a reasonable time with a reasonable amount of computing power, for Google maybe, but not
actually
absurd,
I read
right?
up. I don't think they used some tremendous amount.
No, they didn't.
Yeah, I mean AlphaGo, for example, it was contained in a laptop.
Yeah,
that's what they took.
It's
crazy.
Yeah. No, I mean, well, their TensorFlow chip is also pretty cool. So, but credit to them. Anyway, but again, without that, I guess what I'm saying is that the fact that worked is interesting. Now, are there constraints? Are there proteins that are sufficiently long that would not work? Maybe, but they tried it on pretty long stuff. I mean,
so I was going to ask you, I didn't look into this. So there were some proteins that were pretty large then?
Yeah.
That of the 5 that they did, because that was the...
I mean, you have to remember, it wasn't just... The 5 were the ones they submitted. And I don't know how long those were. But in the training set were long proteins. Right?
And yeah, I suppose that's true. They probably got a lot of right answers in the training
Yeah,
sets too,
of
and
course.
then the
Yeah,
five
yeah.
were the ones that were just offered in the competition.
Yeah, that was just a competition.
So would you say that then they've solved the problem for even enormously long proteins, or do you think that maybe there's some computational barrier that comes up when a protein reaches a certain length?
As far as I understand what they've done, because they've cropped it in certain ways, that means that for the purposes of competition, for the purposes of computation, they limit each computation so that it's not like crazy, right? But because it seems to have worked, it means that you can just do that for any length, right? So it's not obvious that it would work, but after the fact, since it did work, now you can say, yeah, it seems like it probably would work for any length, right? I mean, maybe there's some rare protein out there that it wouldn't work for, but it seems like it's a general solution.
Did they use proteins that cross-linked with each other? Like, say, insulin is a kind of
Correct.
two proteins linked together by disulfide bonds. So that's even more complicated. I wonder if it's an order of magnitude.
I don't know if there was any cross-linking, though. That was actually what it was. So that was the next point I was going to make, is that the same method can be used, right? So certainly for cross-linking, that's one aspect, but even more so for, if you have existing two 3D models and you say, hey, will these two things bind? Well, again, it's a shape problem, it's a distance problem, it's an angle problem. So the same solution set will work for pre-existing tertiary structure. So I know the structure of this protein, I know the structure of this, this antibody, I know the structure of this virus. Will they interact? And this can give you an answer because it'll actually say, yes, it'll come within this distance or not, right? And so that idea of binding is also not trivial. Now you're extrapolating the problem, but the same solution that allowed you to fold it also allows you to figure out if these two things are going to come in, because distance was one of their main things, are these two amino acids going to be close to each other? That was the distance problem that they were solving. And so since they were able to say yes or no to that, it should also be applicable to outside the protein. If the protein will interact with another protein, form a disulfide bond, will it interact with an antibody or a virus or whatever, you can start to solve that exact problem, right? Because within, I think it's like within 8 angstroms was their cutoff.
Yeah.
And if it's within 8 angstroms, I mean, like you should have a chemical bond, right, of some kind. And so you're indicating that there is some kind of whether it's hydrogen bonding or whatever it is. Van der Waals
forces, or electrostatic charges, or
whatever it is. There's some sort of interaction happening to keep it at that distance, because otherwise the electrons would repel from each other.
Yeah.
Right. So unless you form something semi-stable, you're not going to get within that distance.
So 2 follow-up questions. Do you think Alphafold is going to make this algorithm, quote-unquote algorithm, to just give it a broad term, public? Are they just going to release this? Are they going to release the Kraken? Because you could argue they're sitting on $1 trillion technology right now. If they have the... now that they've shown this as proof of concept, there's probably going to be other AI teams around the world.
They've already released it.
They have, so...
Well, not Alphafold itself, but once you say that we successfully reduced the problem to angles and distances, and that was enough to do this, then every other AI team in the world can work on
Gotcha.
You've already told them the solution.
So this is not going to be the kind of exclusive domain of Alphafold or Google, for example, for that matter. Or,
the challenge now, neural nets are now so commonplace. The challenge is not running it through a neural net. The databases are public of their training set, right? All of those are public. It's all public information. The key insight was the engineers who said, yeah, we can reduce this down to distances and angles, and that's enough to solve the problem, right? And we don't have to do it we don't have to do it for the whole protein at once. We can segment and still get the result.
Yeah, I see.
Right? Once you've proven that, then every ATI team in the world can work on optimizing from 90 to 98 to 99%.
Yeah. And if the computation power required is not a lot, which it seems like it's not, it's contained in most modern laptops will be able to handle it. you could have a high school student start to
No,
work
I mean, they
on-
did use a TensorFlow chip, so not most modern laptops.
Most
but-
universities, colleges
Yes,
will be
yeah,
able
yeah,
to
by
afford
far.
most, and every government will be able.
And
to
you can buy a
fund.
TensorFlow chip from what I know. So it's not like it's out of reach even of the public. I'm just, most people don't have it.
So this technology essentially is going to be disseminated worldwide in a few years. Everyone will be solving their own. Actually, if you think about it, you really only need one company. They can just pop these shit out. Pop these 3D structures out like 20 a minute if they wanted to. There's we have the
I don't know if
primary
it's that
structures.
fast, but okay, yeah.
Well, theoretically.
Sure, sure.
It could be that we have mapped out all the 180 million proteins that are out there known to biology. Have them all mapped out in in 12 months? It's a computation. It's a computing power, right? A problem. So if you have enough computing power, you can essentially have them mapped out.
The implications of this are actually quite staggering because you mentioned like 20 or 30,000 that we know. And you mentioned also X-ray crystallography and electron microscopy.
To get that, 2-3 decades.
More than two.
Yeah.
3, like 4 decades.
Yeah. Perhaps
4
as
decades,
long as that,
something like that. It's been a long time, right, to get 20 or 30,000.
Yeah.
And X-ray crystallography is very hit and miss. You have it's very technical. It's like half black art, right? Like trying to get the 3D structure correct is really, really difficult, right? So that's why. But we know the primary structure of
everything.
Yeah,
because
right?
So that
because
I guess
you can and
that's
why?
good,
because you can denature it.
Yes. Well,
right?
not only that, but
it's
a good area to explain also that the primary structure is really just mapped one-to-one with DNA sequence, right? So that's maybe something we didn't explain is you've got a DNA sequence. That DNA sequence directly maps onto the primary structure, and of course we have DNA sequences.
That's not exactly true. I mean, because there's post-processing and there's
well, the amino acid sequence though maps on one-to-one with the codons in a DNA. It was
no, but after DNA is transcribed to mRNA, there are sequences that are cut out and refused.
You
so
mean
it's
like
not
end sequences? Yeah.
I'm just saying.
Yeah,
so
but we know what those end sequences that are cut out are as well. So
not for.
Always. But in any case, primary sequences are freely available to us, right? And so it'll be relatively trivial. In fact, I was thinking initially. Now we're going to have almost like an intellectual arms race to map out all the 3D structures, but just discussing with you, that might be the problem that's solved within a year or two. Because if it's just a computing power and we have the primary sequences, just turn the thing on, go to sleep, and the next morning you'll have at least 2 million done. Right? So I mean, it's almost trivial. And then I guess maybe I was thinking that the frontier of this development is going to be just to get the mapping out there, right? But now I'm thinking that's just it. That's a solved problem. That's you run the servers for a few days and you've got a few million to work with, and then the next frontier of this science is going to be, well, what do you do with that information? And I guess that's another a good place to start the next subject.
No, that's where the excitement is, right?
Here we go, Zane. Here is a 3D structure. Now, what do you do with this exactly? Because that doesn't necessarily mean we have unlocked biology either, right? Because now we're looking at the 3D structure and it's incredibly complicated, and we're like, what the hell does this thing do?
Well, but I mean, no. So again, like I think the same computation method I mentioned, like, can be applied to will this bind with this, right? Will X bind with Z, right? So you end up with some really interesting possibilities for, figuring. So for instance, simple examples, right? So strep throat. But this is one of my favorite examples, actually. So why do we treat strep throat? Right. Something we've lost sight of is actually most cases of strep throat are self-limited. If you don't take antibiotics for strep throat, 99% of them get better on their own. Right.
Is it mitral valve?
It's disease? Mitral valve. Yeah. Right. Is that if you let strep throat just rage, especially in children, yes, the children will get better and everybody will get better. Right. But the antibodies you form against strep throat also cross-react in some cases. Relatively small percentage, but in some cases to your own heart valve, right? And then you end up with heart damage because you have an antibody that binds to strep throat, but also binds your own heart valve, right? And then you end up with rheumatic heart disease and you end up with problems, right? So that's actually why we treat strep throat with antibiotics, right? It's not actually to make people feel better because we're assholes and we don't care whether people feel that. But that's the importance of treating strep throat, is to prevent that binding. Now, if we understand why things bind and what they bind, then I can pull a particular child's antibodies and figure out, is this going to cross-react or not, right? That's a simple, small idea. Same for autoimmune diseases.
But then what do you do about it?
Well, so now that's the next phase, right? We'll get into that. The other possibility is autoimmune diseases, right? Will this antibody bind, you have lupus or you have whatever, like, will this bind this? That's also, that solution is contained in Alphafold's solution, right? So the next step that I think is also the most exciting to me is the possibility of designer proteins, right? Is now you can say, well, I can start creating proteins and now I know how they're going to fold, right? And I know what they're going to bind and what they're not, right? And I have a whole database of existing proteins, right? So I can actually start to create molecules that will interact with the body in reliable ways, right? Right now, all of our drugs are sort of like
machine guns, right?
Yeah, I mean, they're totally non-specific, right? They're just vague, and they're discovered by chance mostly, right? So there's no design or thought that goes into discovering a new drug. I mean, there is, but but it's mostly like trying to figure out, well, is this likely to have an effect on this because this plant makes it, or this whatever? Can I make a version of this plant's substance that will be longer lasting or whatever? So it revolutions the entire concept of drug creation, right? Because now if I want something that binds this enzyme, I can design that and say, hey, I want a protein that binds this enzyme, period, right?
I wonder if you could retrospectively do it, right? So you've got the 3D structure of the thing you want to enzymatically affect, right? You've got that molecule or, a receptor that you want to attach. And then you say, right, what 3D structure would you need to attach it there? You figure out what that 3D structure is, and then you retrograde design the primary structure or figure out what the primary.
I would say that is a separate problem. So I don't know that AlphaFold's current solution includes that automatically, but certainly it's now within reach.
Yeah, it's
right?
within
certainly
reach. If we have all of the 180 million proteins in biology mapped out, we could run the database and see if there's a match out there, right? I mean, like penicillin, where does that come from? It comes from a fungus, right? So maybe there's some cantaloupe urine has a protein that cures cancer. Who knows, right?
No. Chuck Norris's tears cure cancer. Okay.
Chuck Norris's tears.
Yeah. The problem is Chuck Norris doesn't cry.
That's right. Don't know how we're going to solve that problem.
Yeah,
but that's like all of the recipes, all the antidotes that we find are already present in nature, and we just blindly stumble across them, right? Alexander Fleming sneezed on a Petri dish, and suddenly we have penicillin, right? I mean, it was a complete fluke, and it opened up how many more fronts are we going to open up of like protein biology now, where we are now starting to use proteins in an intentional way, and we have great amount of control with the granularity at which we deploy these proteins. I mean, it's an incredibly exciting frontier.
Incredibly. I mean, and I don't know, this also appeals perhaps to my aesthetic of science, which is always a tricky thing, but this is also some source of our arguments over the years in different ways. I'm rereading Dune, actually. So one of the quotes I came across that perfectly encapsulates this is the claim is that he says a process cannot be understood by stopping it. Understanding must flow with the process. And so much of our science, until recently, is reductionist. Is we try to understand physics by literally, like I love the comparison, particle colliders like taking two highly, delicate Swiss watches and just smashing them together and trying to watch the explosion carefully enough to figure out how they work. Yeah. Right. And so much of our science, again, the elucidation of the primary structure of proteins is basically like, hey, let's stop this. Let's pull this out into a string and then kind of understand linearly how this string is put together. And then for 40 years, we have no idea what to do with that information, right? Okay, how do we put all this into a thing? Now we're starting, I think, or at least this is an example of our understanding starting to flow with biology. Is like, okay, if we're going to design proteins, right, we have to design it according to rules that we don't know why they fold this way. By the way, there's no claim of understanding why.
Yeah, that's right.
It is purely a probability distribution.
Yeah, that's right.
Right?
Yeah.
Based on existing models, we're able to create a probability function for how this thing is probably going to fold. But I don't need to know why. Right? And that's exactly my point, is we're not trying to get into the physics of exactly how these chemical bonds form and this. We don't need to model any of that. Yeah. Skip all of it. This is how it's going to fold. Now let's use that understanding to move forward, right? And I think that that's an example of, hey, let's not try and stop the whole thing and model every electron, right? Let's just move forward. Let's kind of go with the way biology works, right? Which is that proteins do fold. And they fold in predictable ways. And now we've got some tools to figure out to predict what that is. And now let's try and build on that understanding, right? So I don't know. I think that is also a little bit of to me, a sea change in some sense of not trying to get into the nitty gritty of, you don't have to solve physics in order to solve biology, right? Yeah, and so that kind of says, okay, hey, we're going to skip that by using a computational model that seems to be effective enough that we're not now reducing everything to, well, until we have a quantum theory of gravity, we're not going to be able to. I mean, like, that's an unreasonable expectation. We're sort of short-circuiting that and starting to let our understanding flow with biology and kind of like grow these new, maybe designer proteins or whatever it is that we're doing next on the basis of incomplete understanding, right? But good enough.
I'm actually surprised by your optimism, and that's just because I know you as well as I do. I was, we didn't talk about this subject much at length before we started recording, so I was prepared for you to start shitting all over this, discovery, telling me that, well, no, there's going to be, we're dealing with incredibly complex systems, and you're going to throw a protein in there that you've designed that's going to cause, us to grow horns.
Oh, it will. Don't get me wrong.
Okay,
no.
There you go.
Yeah. But it'll probably be worth doing nonetheless, right? And I think that I think the reason I'm more excited about this is we're not trying to get a granular understanding. We're skipping that. And that to me is why it's probably going to work better, right? Our approaches to things like genetics, our approaches to things like we're trying to do these like micro understandings of stuff that has complex. We're not embracing the complexity. This just embrace the complexity. Hey, it's fucking complicated. It folds in some weird way. No idea why. But maybe we don't need to know why, right?
Well, so
that to me is
the complexity that you sometimes push back at in our conversations is that you are introducing a protein that is going to maybe enzymatically cause a reaction in this specific domain, but in an incredibly complex and crowded protein field, which is the human protoplasm, you have no idea what else it's going to do. Mm-hmm. And so you are, literally playing with fire, right? We're flying too close to the sun here, and we are going to go ahead and introduce designer proteins to solve cancer, and then maybe we'll cause some other horrific side effect that will, pass down to generations, though.
That's going to happen.
So where's your pessimism? I guess I'm reaching
here. That's totally going to happen, right? That's totally going to happen, right? I think
because you really whenever we talk about say genetic engineering, I mean you just rain like
because we don't understand it. We don't understand it, and we're trying to understand it at too fine a level of detail. We're hinging our success on too fine a level of understanding, right? So that's why I always say is like we're going to get sickle cell, we're going to get cystic fibrosis. We're going to get some of the rare genetic mutations that are point mutations because we understand them, right? And I absolutely think our genetic engineering efforts will pay off for those diseases, right?
Well, would protein engineering help with those problems? So specifically, cystic fibrosis would be a great example of a potential designer protein. And it would actually be a very, very good example, because cystic fibrosis, although it also affects the pancreas, primarily affects the cilia in the the mucosal layer, and you could very easily create a coronavirus, say, that injects a particular RNA code that then codes for a protein that I forget what the
it's an ion channel.
Ion channel. Okay, so that then codes for that ion channel. And
yeah,
perhaps,
you could. I mean, you certainly could. Like again, cystic fibrosis. There are multiple variants, but the most common variant of cystic fibrosis is, I think, if it's the CF gene I think it's on chromosome 7, right? So we know which gene it is. Things like CRISPR or other gene editing technologies, if you catch a kid young enough, like they're turning over their cells so fast that if you can actually just you change that one amino or that one base pair, and you basically are done, right? And I think stuff like that is going to be solvable, right? With pretty quickly overall. I think we're gonna get that initial success. You're gonna hear a lot about it. It's gonna be big hype. Hey, we've done this. Look how much more is coming, and nothing's gonna come. Right? This is different. Right. Protein folding is different because we're not trying to solve it granularly. We solved a general problem of folding, right? That doesn't have, like, DNA has to be transcoded to RNA. There are epigenetic effects. There's multiple layers. Like, there are other proteins that come and snip the RNA. Then it makes a protein. That protein gets snipped and then restructured, right? There's like 12 layers, I see what you mean, Whereas this is the final engine
solution.
Yeah, downstream solution.
So,
right? And it's much simpler that way.
Yeah, that's right.
Right.
Yeah.
So now you're not trying to futz around like in a black box.
Yeah.
You're just saying,
the black box being the interplay between genetics and
epigenetics and all of that. Yeah, you're skipping all of it. You're just going straight to the end product, and your solution there is not contingent on understanding other things, right? It's a general solution for the way this folds, and then from there you can say the way that binds. Now, there's going to be a separate problem, is like, are you going to catalyze a particular reaction so simple things like antibody binding, right? That's going to come first, because just binding it is all you're looking for. You're just looking for that chemical bind. Things like more of the protein actions that we think of in terms of like the Krebs cycle, or things where the protein has to do something after it binds. It's gonna take another layer, right? So still some questions there, but just the binding itself is super exciting. Yeah. And that's all, again, I think that's incorporated in the solution they came up with for how this folds. They've also solved that problem, because if it comes close enough, it forms a bond. Yeah. Right.
So could you imagine, say, if we had a specific kind of cancer, right? Say pancreatic cancer, right? One of the most devastating, stage 4 pancreatic cancer, right? So say we are able to find the genetics of that particular cancer. We get a biopsy, and then we find out, well, this is the genome for the pancreatic cancer, and find out what proteins, or we look for some membrane proteins in the cells of that cancer, and we say, all right, can we create an antibody that complements some member proteins, right? And then create a designer antibody that labels and attaches only to the cancer cells. So is that a conceivable pathway, you feel?
Conceivable, for sure. I think there's some trickiness, and it'll vary cancer to cancer.
Yeah, of course.
But meaning even whether it works, cancer to cancer, in that, some cancers may have unique proteins that are featured, right, that are unique enough to bind to, right? Others may not, and I don't know the answer to that, but often some cancer, I don't know enough. I'm not an oncologist, so but obviously cancer is the hallmark of cancer is unrestrained growth. Yeah. And so designating a cell as cancerous versus non-cancerous is actually not trivial. Yeah. Right. But if you could, then certainly, and I guess that's the point, is that if you could come up with a chemical assay to diagnose the cancer, then the treatment is already built, or, you kind of have this idea is that now we can build an antibody that's going to bind that, right? We do have natural killer cells. We have anti-cancer, like we've all gotten cancer already, right? Course. And our immune system has anti-cancer capabilities. We attack those cells, we kill them, whatever. Now we have the option to say, oh, okay, hey, you missed this one. Now it's stage 4. Now it's gone, whatever. But if we can find something that is unique to those cancer cells to bind to, right, then we can actually, again, this is flowing with, I'm not trying to solve cancer. I'm just trying to tell your body you have cancer, right? And then your immune system take care of it.
Yeah.
Right?
I mean, labeling is, say, one example, right?
Yeah,
but
yeah.
I mean, another would be if a particular cancer cell, and every cancer would be different. You'd have to catalog the different cancers and the genetics of them all. But if a certain cancer cell had enzymes, a specific enzyme that wasn't present anywhere in the body, you decide to go ahead and short circuit that enzyme with an anti-enzyme, say, right? You wouldn't have to be a labeling with the immune system. It would just be you go ahead and infect this cancer with proteins that basically short circuit its metabolism, right? And then it just sort of ends up eating itself up, or just commits apoptosis or whatever. In fact, that would be another thing. How do you could trigger apoptosis? In certain cancers if you knew exactly what protein switches you were aiming for. So theoretically, of course, I mean, even I don't know that much about the genetics of cancer and how the therapy would work on a granular level, but would you agree that this at least opens a possibility for the beginning of the end for of cancer? Is this the
I would have to know
is this the
more
theoretical
about,
foundation that we needed?
I certainly think it could. I don't know. I don't know. I'm a lot less optimistic in these ways with you, but actually, for this particular thing, it is so revolutionary that it does make me optimistic, because it is. I really feel like with DNA and genetics and all this stuff, we're futzing around in the dark and we pretend we know more than we do. This is a meaningful understanding of, hey, these are the tools that our bodies use to do stuff. Now we're going to understand those tools. Now we're going to be able to create them. Now we're going to be able to create new tools, modify them, look at the tools that plants use, that other animals use. We've got, this is sort of like, opening up an entire arena of speaking the language. Yeah. Right. And I think that's maybe the difference. Is I really feel like this solution is us starting to, at least it's sort of the Rosetta Stone of understanding the language, right? And we don't have that for genetics. We're not even close to that with genetics, right? We have, I think, my personal opinion, we have no idea what that language is. We've barely even got the idea that there are particular letters, maybe, right? This is a huge step. Like now we're actually in the race. Yeah, right? We know that proteins are enzymes, they're ion pumps, they're transmembrane receptors, they're antibodies. We know how those mechanisms work, right? And now we're like, all right, what if we designed our own, Yep. And that's just in the field of biology. I mean, in the field of human biology, right? I mean, there's also... And there will be unintended effects. We're going to try this on people. No question, there's going to be weird black swan stuff, and there's going to be unintended effects. But in, say, 50 years of this development, though, this should be an incredibly, incredibly powerful, tool. And in fact, one way of thinking about it is the age of, the metallurgy, right? When we were able to manipulate metals, to create, stronger structures. This is kind of that age, where this is the protein age. I mean, it really is. It explodes a frontier. There's going to be now protein robotic science, perhaps, right? Where people sit down and figure out how to design a protein to do something, like digest plastic, for example. Yep. That's the one that has already been talked about. Sure. Because that's literally just an engineering problem. And now you've got the tools you need. No, but it goes further than that, right? So, there are fungi that digest plastic. Right. How are they doing it? Exactly. How are they doing it? They're doing it with the protein. I guarantee you that. Right, exactly. Right? We know they're doing it with the protein. But how will we replicate that? Right. How do we know? How do we know which? Yeah. Right? Now you can all do these it problems are solved. Now you don't need a fungi to do it. Now you don't need a fungi, right? You just get an industrial-sized vat. You put a bunch of that protein in there. The same way we make other proteins, right? You tell yeast to do it. Yeah. Right? And in that regard, actually, so that's an aspect of genetics, by the way, is that we don't know in the forward direction how our genes translate to proteins. But we do know how to create fake DNA or fake RNA that will reliably create certain proteins. That's a solved problem. Yeah. right?
Recombinant DNA technology.
Recombinant DNA, exactly. And so, okay, we may not understand our own genes because that's more complicated. There's epigenetic phenomena, but you can reverse engineer a plasmid of DNA that will reliably make a particular protein. You take that plasmid, plug it into a yeast, the whole vat will start making that protein, and you can ingest as much plastic as you want.
Yeah. I guess the cutting edge of that iteration
I mean, put it in a glass vat. Don't get me wrong. It's in a plastic vat.
Don't
sorry.
put it in a plastic bag.
Yeah.
The cutting edge of that iteration is the mRNA vaccine, right? I mean, you're basically engineering the body this time to make a protein, and who knows what's going to happen? I guess we'll find out, soon enough.
Well, but that's exactly the point, and that's why I'm more enthusiastic about the mRNA vaccine than I are about other genetic technologies or whatever, is that, again, we're not trying to understand the natural thing there with some sort of like linear understanding. We took our linear understanding, our simplistic, basic understanding, and we reverse engineered a protein. That we know how to do.
Yeah.
That's not rocket science, right? Like we already knew what the protein was, and we just made an mRNA that makes that protein.
So if Alzheimer's, right, is because of the accumulation of a certain, aberrant protein, right? What is the name of the protein?
Tau protein.
Yeah, the tau protein, exactly. If that hypothesis, and I'm not saying that, that's a settled question, but or other diseases that are amyloidosis is a good example. If those are basically just protein deposition diseases, let's call them, right? Sure. Would creating an enzyme to dissolve that be the
Maybe.
solution?
I mean, that's a great example of where I think these unexplained effects may come into effect, and we'll have to try stuff like that out. Yeah. So they've done studies in mice specifically with Alzheimer's. They've given them. We actually have the enzymes that destroy the tau protein tangles, right? And consistently they make it worse. So the tau proteins get disintegrated, and the Alzheimer or the brain degeneration gets worse. So again, you have to make sure you've understood the disease correctly, and that's one of I read some recent articles on Alzheimer's, is that actually the tau proteins may be protective against whatever's really going on.
Right. Gotcha.
Right. So it's a symptom, or a marker, not a contributor. But point is, yes, if you accurately understand the problem, then on some level, and maybe that's the exciting thing, is that there are a lot of problems that we don't understand yet. But once we understand a problem, we've got the solution built.
Yeah.
Right.
And in fact, we don't actually even have to understand it. We can try different solutions, see what worked, and then, retrospective. I mean, not on humans. Not on humans. I mean,
on humans. What are you talking about? Yeah. Just give it to them. Just inject them.
Like you could do it on animal studies, for example, right?
Ethics,
if
whatever.
You come up with a protein that has a dramatic effect, then that unlocks the potential, like, oh, maybe this is the way this direction is where we need to go.
Yeah.
Now, for the first time, we actually have those nano tools where we can run those experiments. We're not just carpet bombing them with antibiotic-like, chemical substances.
Actually, antibiotics is a great example as well, right? And we mentioned this briefly in terms of the mRNA vaccine and stuff like that, right? But, on the horizon, we all know as physicians that we are teetering on the edge of a post-antibiotic era, right? Because we've used antibiotics for agriculture at deliberately subtherapeutic levels, which basically just trains bacteria to be resistant, right? We've given Z-Packs for every fucking cold, right?
Wait, are you not supposed to do that? Z-Pack and steroids.
Z-Pack and steroids. Kill me. But yeah, I mean, and we've gotten this idea that, you go to the doctor to get an antibiotic. That's part of the unholy pact, I guess, that insurance has forced us into. And for all these reasons, I mean, like there are strains of gonorrhea that are resistant to all known antibiotics. Those exist. Yeah. And that's a scary thing. Yeah. Right? I mean, there's colistin-resistant enterocon. And colistin is basically IV bleach. And there are bacteria that are resistant, right? And yeah, I mean, we are teetering on the edge of TB, a disease that we have not seen in this country at prevalent levels for a long, long time. But you get enough multi-drug resistant strains and you can't fight it anymore, and it comes roaring back. And you think quarantine for coronavirus is bad. You wait until you're locked in a TB ward, and the state basically like put you in prison to feed you like random stuff. But this offers a way out because all of those things have, the same assay we use to detect, do you have this disease? We now can design a protein to bind it.
Yeah.
Right. And once you bind, you've designed an antibody that will bind it. I mean, that's how all these assays work in some sense. Is like, hey, the rapid strep test, it binds an antigen on strep, right? So we already have that technology. Now we can create designer antibodies, right?
Yeah, designer antibodies is. I mean, there are multiple
and they're temporary.
ways.
They're going to be broken down, right? So it's not like we're incorporating something into your genome that's going to stick with you. We give you a protein, maybe we give you a couple dose, maybe we give you a dose every day. Who knows, how it works out until you've cleared that infection, right? But now, again, we're not doing this weird, as you say, carpet bombing thing where, Calliston's IV bleach, I mean, like we're poisoning you, hoping that you're stronger than your infection and you will live longer after, than the poison's not strong enough to kill you, but it's strong enough to kill the bacteria or the virus, right?
Or the cancer.
Not the virus, but the bacteria. Yeah. Oh my God. Yeah. Chemo is literally that, right? It's like literally just expecting you to have bigger, deeper resources than the cancer does, right? And it's very, very photo finish right? Most of the time.
Yeah, a lot of the times it can be.
But yeah, the potential here is that we're not doing that anymore. The potential here is that we're actually moving to
a very granular, fine-tuned targeting mechanism.
Because we're not trying to use robots to do something in the body. We're using the natural language, right? We're doing this in the language that the natural world uses instead of trying to do something mechanical, right? Which is a different domain and it doesn't work. And inevitably it becomes very rough-handed. It's very you and I've talked about this before, I mean, like just how brutal medicine still is in so many ways. There's still so many aspects of medicine that are just like, I don't know, rough solutions in a lot of ways because we don't have this language. We're not talking the same language as the natural environment. And as a result, we're applying mechanical solutions to natural problems. And, it's not going to be a granular sort of elegant answer. It's going to be a very inelegant answer.
Do you think the team over at DeepMind is going to win a Nobel Prize anytime soon?
Hadn't thought about it.
I think, they're going to. I don't know what they're going to get. Is it for biology or chemistry, or I'm not even sure. But they have a Nobel Prize headed their way. I think it's inevitable.
Yeah.
I mean,
I don't know. It's interesting. Yeah, maybe. It depends a little on how. Because what they did was they developed a computational method, right? So it depends a little on how old the people are at the Nobel committee.
No, it doesn't. That doesn't matter at all.
I'm being a little facetious, but the reason I say is it doesn't fit neatly into a biology box, right? It does. I mean, it is a biological problem, obviously, but it's a computational method applied to biology, and so
they'll find some Nobel Prize to give them, though. It's I feel like this is about on par with.
It's a truly revolutionary thing. No
it's
question.
about on par with the genetic sequencing that happened. The DNA sequencing of the human genome that occurred last century.
I think it's far beyond that.
Oh wow! Okay, I mean, I thought that
far
was
beyond.
Yeah.
I think that's horseshit.
In terms of where it can lead us, yes, yeah. But I mean, like the encoding,
the whole human genome thing was like
hyperbole. You think? Or?
Not just hyperbole, but it was like, oh, look at this feather in my hat. I'm so fancy. Like it doesn't have any relevance to anything, right?
I think
it's a pure vanity project.
Yeah, I think maybe
pure
it's a little
vanity.
bit more than a vanity project.
No,
but,
pure vanity project.
But yeah, no, I agree. I mean, this is at least on par.
Yeah,
with,
you know how I'm negative towards
I mean,
but yeah,
this is probably
I think it's far beyond
the biggest breakthrough in medicine or biology in our lifetimes. Honestly, in our lifetimes, there's nothing bigger. I mean, no one's talking about it in the news because they don't understand how huge this is because we're all concerned with whatever stupid. Is there a pandemic or something going
Yeah,
on?
I don't know,
heard something about
I heard
that.
something.
But that might be that's something.
Yeah,
that people are
no,
worried about.
it is interesting how this is one of those examples where, we've heard for so long about this genetic breakthrough or that genetic breakthrough, and it's all doesn't lead anywhere, right? And now I think we have a genuine breakthrough, and nobody's talking about it, right?
we have I mean, I don't think we're going to see the fruits of this in terms of designer proteins for at least another half decade at the very least, I think.
You're so optimistic on your timelines. You're such an optimist on your timelines.
No, the reason I say that is because there's going to be multiple. I mean, there's going to be an arms race to find that next thing that cures this next disease. I mean, you are unleashing the power of capitalism, right? Universities are not going to do this as an intellectual vanity project, right? This is going to be like, if we can design a protein that will cure this disease, and that's $1 trillion market, you're going to have pharmaceutical companies all over this.
Yeah.
And so this is basically now you give this tool to capitalism, and you say, all right, if you can figure this out, you can cure whatever, X disease. I mean, definitely pandemics should be a thing of the past, like if another coronavirus comes, you sequence it, you come up with a reasonable target, you spit out, the protein you need, and then you just, send it in an email to your local protein lab. I suppose that's going to be a thing one day. And then, that's the end of that story.